Okay, so welcome back to this next video in which we are discussing ion channels and cancer. Okay, right. So we're currently in the process of discussing ion channels. Okay, so we're looking at the potassium channels, and we've seen that there are four main families of potassium channels. We've looked now in detail at the voltage-gated potassium channels. We're going to continue that in detailed study of the different families of potassium channels, and the next one up is the calcium-activated potassium channels. So let's go there now. Okay, right. So calcium-activated potassium channels, KCA. Right, so calcium-activated potassium channels have a very similar structure to voltage-gated potassium channels in that they too are tetramers. So if once again I draw this in the plasma membrane, so this is the outer leaflet, this is the inner leaflet. Again, it's a tetramer of four separate proteins that have come together to form the whole calcium-activated potassium channel, like so. And this will be sitting in the plasma membrane. So here it is. Right, so very similar structure to the voltage-gated potassium channels. In fact, some of the subunits of calcium-activated potassium channels have the same membrane-spanning topology as the voltage-gated potassium channels. Okay, so I'm going to tell you about the three main families of calcium-activated potassium channels. There are two other families, although what their role is much less well understood. So I'm going to tell you about the three mainstream families of calcium-activated potassium channels, which are the only ones we're going to see in this video. Okay, so the three main families then are the KCA1, the calcium-activated potassium channels family number 1. Then family number 2 of the calcium-activated potassium channels, and then KCA3 as well. So these are the three main families. Now, let me tell you about the members of these families then. So the KCA1 family only has one member, which is KCA 1.1. Okay, the KCA2 family has three members, which are KCA 2.1, KCA 2.2, and KCA 2.3. Okay, and finally KCA3 again only has one member, which is KCA 3.1. Right, okay, now these then are the subunits which make up the mainstream calcium-activated potassium channels. So let me show you now their membrane-spanning topology. Okay, so for the KCA2s and the KCA3s, it's exactly like it was for the voltage-gated potassium channels. Okay, so again you have the 6 membrane-spanning alpha helices with a p-loop in between the 5th and the 6th. Okay, now I'm not exaggerating the S4, S5 linker the way I did previously. Okay, so here is the exact same membrane-spanning topology as the voltage-gated potassium channels that we've seen previously. So all of those four proteins have that same membrane-spanning topology. However, KCA 1.1 is slightly different. KCA 1.1's membrane-spanning topology looks like so. It actually has the amino terminus extracellularly, and then it's exactly the same. Then you have the 6 remaining membrane-spanning alpha helices with a p-loop in between the 5th and the 6th. Okay, so it has this initial first membrane-spanning alpha helix that the other ones do not. Okay, so this is how it's different from the others. Okay, and that first membrane-spanning alpha helix in KCA 1.1 is known as the S0 membrane-spanning alpha helix, and then the name rings exactly the same: S1, S2, S3, S4, S5, P, and S6. Okay, right, so again, these can associate up into tetramers. Now the main types of tetramers that we are going to see are going to be homotetramers again. So let me tell you about the homotetrameric calcium-activated potassium channels. Okay, so if you take 4 KCA 1.1s together and put them together to make a calcium-activated potassium channel tetramer, okay, what that channel is now called is a BK channel. Okay, now what does this stand for? This stands for big conductance calcium-activated potassium channel. Okay, so this is a big conductance calcium activated potassium channel. Now, to emphasize the fact that it is a calcium activated potassium channel, some people will put a subscript Ca after the K. So some people will denote this BKCA. That's the same thing. That's just to emphasize the fact that it is a calcium activated potassium channel. Okay, but if you just see BK, that means the big conductance calcium activated potassium channel. Okay, right, so this is a channel which can be activated by intracellular calcium, and when it opens, its conductance is very high, and that means how much potassium it's capable of conducting through it is very high. Okay, now, if you make calcium activated potassium channels from KCA2 subunits, you get what's known as a small conductance potassium channel, and you can either make homotetrameric channels or you can make heterotetrameric channels. So an SK channel means a small conductance calcium activated potassium channel, so I'll just put the small conductance portion, and then I might just put KCA for calcium activated potassium channel. Okay, so again, some people will denote this, and I'll just move this up. So some people will denote the small conductance calcium activated potassium channel as SKCA to emphasize more that this is a calcium activated potassium channel. Now I will stress again that SK, small conductance calcium activated potassium channels, they can either be homotetramers or heterotetramers. All it means is that the 4 subunits that you use are from the KCA2 family. Okay, now the ones that we're going to see are going to be homotetramers. Okay, so let me just tell you a little bit more about the nomenclature for the homotetrameric small conductance calcium activated potassium channels. So you can also see the nomenclature SK1, SK2, and SK3, and these refer to the homotetramers of KCA 2.1, KCA 2.2, and KCA 2.3 respectively. So this is a KCA 2.1 homotetramer. Okay, so you take 4 KCA 2.1 subunits, put them together, and that makes an SK1 channel. This one is the KCA 2.2 homotetramer, and this one is the KCA 2.3 homotetramer. Okay, so just repeat that word there and there. Okay, right, so that's a little bit more nomenclature there. And when these open, again, they're activated to open by intracellular calcium. When these open, they form a potassium channel which has quite a small conductance, so it's much less good at conducting potassium ions through it than is the big conductance potassium channel. Okay, right. Now we'll finally go on to the KCA3 family. Okay, if you make a homotetramer of KCA 3.1 subunits, that's what's known as an intermediate conductance calcium activated potassium channel. So an IK channel, or indeed you can also denote it IKCA to emphasize the fact that it is an intermediate conductance calcium activated potassium channel. Okay, so again, that's always a homotetramer, just like the big conductance calcium activated potassium channel. Okay, and these are going to be important later on. We will see a lot, particularly of the intermediate conductance calcium activated potassium channel. Okay, right. So next family of potassium channels that I want to move on to is the inward rectifying potassium channels, the IRKs. Okay, and I want to explain to you why they are inward rectifiers. Why do they prefer to let potassium move through? And I said earlier that it's nothing to do with the channel actually preferring to let potassium move in one direction. Okay, in fact, it's to do with the fact that it's more likely to be blocked under conditions when potassium would be moving out than it is under conditions when potassium would be moving in. Okay, right. So let's have a look at the structure of these inward rectifier potassium channels, which are also called KIR. Okay, so these again are tetrameric. Okay, they're made of 4 separate subunits. So again, I'll draw it like so. Here is the pore in the center, and you have 4 separate proteins making up the 4 tetramer. Okay, now, same protocol as before. Let's take out one of these subunits and have a look at its membrane spanning topology. Okay, now it's very, very simple membrane spanning topology, and this means that these inward rectifying potassium channels really do just look like what I've shown you here. Okay, so the membrane spanning topology of an inward rectifying potassium channel subunit looks like so. It has one membrane spanning alpha helix, then a p-loop, and then the second membrane spanning alpha helix, and that's it. Okay, so this first membrane spanning alpha helix is called TM1, and the second membrane spanning alpha helix is called TM2 for transmembrane domain 2. Okay, and then you've got the p-loop in between. So this basically is just like the pore-forming domain that we saw of the voltage-gated potassium channels and also of the calcium-activated potassium channels. Okay, right, so let me just highlight these different portions up. So here's TM1 in red and TM2 in red, and then we've got p-loop in purple. So you put 4 of these together to make a tetramer, and that's your inward rectifier potassium channel. Okay, right. So now let's talk about how many different families there are of inward rectifying potassium channels. So inward rectifying potassium channels are split into 7 families. Okay, and again it's the same nice nomenclature. It's KIR1 to KIR7. So the inward rectifying potassium channel family 1 to inward rectifier potassium channel family 7. Okay, and again the members of the family are then denoted KIR, so let's say we're talking about the first family, it would be KIR1.X then. Okay, so it's the same nomenclature as always. Okay, and again, you can form homotetramers and heterotetramers of inward rectifier potassium channel subunits. Okay, so the big thing that I want to now talk about is why are these channels inward rectifiers? Okay, why do they prefer to let potassium move in than out? Okay, well, let's talk a little bit about the Nernst potential for potassium, because that's the essential thing for understanding this. Okay, so let's draw our plasma membrane here. Okay, in fact it might also be helpful to, actually no, we'll keep it like this. Okay, so if we have our plasma membrane here, let's talk about the intracellular and extracellular concentrations of potassium. So the extracellular concentration of potassium is generally around 4 millimolar, and the intracellular concentration of potassium is around 155 millimolar. Okay, so let's initially imagine that there is no electrical potential difference across the cell membrane, and we were to open a potassium channel. Which direction would we get a net movement of potassium in? Okay, well it's obvious. We'd get potassium moving outwards because the concentration gradient is there that would be moving the potassium ions out. Okay, so that's assuming that there is no electrical potential difference across the cell membrane, i.e. both sides have the same electrical potential. Everything is neutral. Okay, you get potassium moving out. However, that's not the case usually in cells. In cells, we usually have an electrical potential difference across the cell membrane. There is a difference in the electrical potential extracellularly compared to the electrical potential intracellularly. Okay, and this electrical potential difference is usually somewhere around negative 65 millivolts. Okay, and what that means is that the electrical potential intracellularly, okay, which we might denote capital E for electrical potential and then subscripted I for intracellularly, is 65 millivolts lower than the electrical potential extracellularly, which will have capital E for electrical potential and then subscripted E for extracellularly. Okay, so if you move across the cell membrane from the extracellular compartment into the intracellular compartment, the electrical potential goes down by 65 millivolts. Okay, right, now what does that mean for the movement of potassium ions across the cell membrane when we open the potassium channel? Well, this complicates the matter hugely from when there was no electrical potential difference, because potassium ions are a positively charged ion. Positively charged ions, as a basic rule of physics, want to go where the electrical potential is lower. Now, which compartment currently has the lower electrical potential? Well, that's the intracellular compartment. Okay, by 65 millivolts, the intracellular electrical potential is lower than the extracellular electrical potential. So the potassium ions are going to want to stay in the cytoplasm because of the electrical gradient across the cell membrane. Okay, so the two are pointing in the opposite direction, basically. The concentration gradient says go out, the electrical gradient says go in. Who wins is the question. Who wins? Which is stronger? Well, at this electrical potential difference of negative 65 millivolts, the concentration gradient wins. It's stronger. So if I draw this, let's have the arrow for the concentration gradient like so. Okay, and then the electrical gradient's arrow is smaller, basically. So now, if you take the net arrow, if you add this arrow onto this arrow like a vector, what you'll then get is a much smaller movement of potassium out. Okay, so note, the electrical potential difference across the cell membrane will hugely decrease the amount of potassium that will move out of the cell compared to what it was when there was no electrical potential difference across the cell membrane. But it's not big enough yet to stop the movement of potassium out. So now I can explain to you what the concept of the Nernst potential for potassium is. Oops, not like that. Okay, I can rescue it. Nernst, so that should be an N, not an H. Nernst potential for potassium, okay, which is often abbreviated down to EK, so people often call this the equilibrium potential for potassium. Okay, so what does this mean? This means what electrical potential difference would you need across the cell membrane in order to completely stop any movement of potassium across the cell membrane when you open this potassium channel? I.e., how big would this electrical potential difference need to be in order for this arrow here to be as big as this blue arrow here? Well, the answer is around negative 85 millivolts. Okay, for these concentrations of potassium that you usually have across the cell membrane. So for this concentration gradient of potassium, the electrical gradient that you need to stop the movement of potassium across the cell membrane would be around negative 85 millivolts, i.e. you need the electrical potential intracellularly to be 85 millivolts lower than the electrical potential extracellularly in order that the potassium ions here do not feel the need to move out anymore because the electrical gradient is strong enough to keep them there despite the concentration gradient. Okay, now, if you then go above this, okay, so if you go to negative 65 millivolts, so a less negative electrical potential difference, i.e. if you're depolarized of this, then of course the electrical gradient is not going to be strong enough to oppose the concentration gradient, and you are still going to get potassium moving out. On the other hand, if you go hyperpolarized of this, if you go to that same negative 95 millivolts, what will happen now? Well, it will actually completely reverse, basically. If you go to negative 95 millivolts, or indeed anything hyperpolarized to negative 85 millivolts, what will actually happen is now the electrical gradient will be stronger than the concentration gradient, so you'll actually get potassium moving in. Okay, right, so that's an essential concept in order to understand how these inward rectifier potassium channels are going to work. So basically, there are certain molecules which can block inward rectifier potassium channels. We'll come back to this and we'll put it together. Now I'm going to go off in a totally different direction, and then I'm going to integrate them together in a moment. Okay, right. So there are certain molecules which can block inward rectifier potassium channels. So if I draw my cell here, let's have our inward rectifier potassium channel here. There are certain molecules that are in the cytoplasm of cells which can block inward rectifier potassium channels. For instance, magnesium ions can block inward rectifier potassium channels. Magnesium ions from the cytoplasm can go into this pore and block it. Okay. Also, another example, which I'm not going to try and write in there, but another example that's in the cytoplasm are molecules known as polyamines, okay, which are basically molecules which have a huge number of amino groups. Okay, now what's the significance of having a huge number of amino groups? Well, amino groups are basic. They accept protons. They become positively charged when they accept protons, and therefore these molecules are generally positively charged. So let me give you a notable example of a polyamine. Okay, whoops, spelt it wrong again, I'm doing badly this morning. A notable example of a polyamine is spermine, okay, which is something to do with sperm, okay. I don't fully know exactly what role it plays in sperm, but it is something to do with sperm, okay, but it is a normal component of the cytoplasm as well. So let me show you the polyamine spermine then. Okay, so here is, it's actually got 4 amino groups, so I'll draw its skeletal structure. So here's the first amino group, then you've got 3 carbons, like so. Then another amino group here, which is a secondary amine group rather than a primary amine group. Then 4 carbons, like so. Then another secondary amino group here. Then 3 carbons, and then a final amino group here. So this is the molecule spermine. Okay, right. Now, what's my point here? Well, basically, these amino groups, they all have a lone pair of electrons. So, if I, for instance, show it on this one here, it has a lone pair of electrons here, and it can accept a proton onto that lone pair of electrons, giving the molecule, in total, a positively charged. Okay, so, positive charge is the important thing here. Okay, magnesium ions are positively charged, spermine is positively charged, and these molecules are capable of coming in to the inward rectifier potassium channel from the cytoplasmic aspect and blocking these inward rectifier potassium channels. Okay, now how does this go together with the concept of the Nernst potential to to produce inward rectification of these potassium channels? Well, the idea is this. Basically, when the electrical potential difference across the cell membrane, I'm going to draw this picture here again. Okay, when the electrical potential difference across the cell membrane is depolarized of the Nernst potential for potassium. So let's say it's negative 65 millivolts at the moment, i.e. it's depolarized of the Nernst potential for potassium, which remember was negative 85 millivolts. That means that potassium will want to move out. Basically, the idea is that when you are depolarized of the depolarized of the Nernst potential for potassium, basically these molecules, spermine and magnesium, are going to block the inward rectifying potassium channels. They're going to go into the potassium channel and block it, and therefore they're going to stop potassium ions from being able to move out. Okay, so basically the idea is that when you are depolarized of the Nernst potential for potassium, magnesium ions and spermine molecules and other polyamines are going to block your inward rectifying potassium channels, and therefore block these channels from being able to conduct potassium ions out of the cytoplasm of the cell. Meanwhile, when you are hyperpolarized of the Nernst potential for potassium, okay, so let me now show hyperpolarized of the Nernst potential. So let's say you've gone down to negative 95 millivolts, okay, then what will happen is these molecules, these magnesium ions and these spermine molecules, they're not going to block the inward rectifying potassium channels when you are hyperpolarized of the Nernst potential for potassium. Now why? Well, they're positively charged molecules, so they're going to feel the electrical potential difference across the cell membrane as well. If we are now down at this more negative electrical potential difference across the cell membrane, then the magnesium ions and the spermine molecules are absolutely going to want to remain in the cytoplasm, where the electrical potential is now negative 95 millivolts compared to the extracellular electrical potential. Okay, and that's going to stop them wanting to get out of the cell, so they're no longer going to be blocking these pores of these inward rectifying potassium channels. So it's all about the magnesium ions and the polyamines having a positive charge and therefore not blocking the inward rectifying potassium channels when the electrical potential difference across the cell membrane is hyperpolarized of the Nernst potential for potassium. And then, of course, these inward rectifying potassium channels will be open, but now they'll be conducting potassium into the cell, okay, and therefore they will be open. So the idea then here is that it's not that the potassium channel prefers to let potassium ions move in compared to out. The idea is that when the electrical potential difference across the cell membrane is hyperpolarized of the Nernst potential for potassium, and therefore potassium ions will want to move in if the channel is unblocked, at that electrical potential difference, the channel will be unblocked because these molecules, the magnesium ions and spermine, are positively charged and therefore are absolutely not going to want to be getting out of the cell at this sort of electrical potential difference. Okay? Whereas, when we are depolarized of the Nernst potential for potassium, the magnesium ions and the spermine molecules aren't feeding the same sort of attraction into the cytoplasm, okay? And therefore they might end up blocking these channels, and therefore they're going to block the movement of potassium through those channels, and at this sort of electrical potential difference, then potassium will be moving out of the cell. So you block the outward movement, but don't block the inward movement. So that's why these potassium channels are called inward rectifier potassium channels, and indeed why they do show the phenomenon of inward rectification. It is not, repeat, it is not because the potassium channels actually prefer to let potassium come in compared to out, although that's what it manifests itself as looking like. Okay, right. So that was a in detail discussion of that. Okay, right, final family of potassium channels then, and then we'll briefly discuss the sodium channels and the calcium channels and the trip channels. Okay, so the next family of potassium channels then are the K2P channels, okay, which are the two-pore domain potassium channels, okay, and I will repeat again that these are not just two-pore potassium channels. If you miss out that word domain, then you have made a ghastly mistake. Okay, two-pore potassium channels are very, very putative things which actually literally have two pores. Okay, two-pore domain potassium channels are channels which are built out of subunits which have two P loops basically in their structure. Okay, right, so let me show you the structure of a two-pore domain potassium channel. Okay, so, once again, they're in the plasma membrane, like so, and this time they are not going to be tetramers. Instead, they are going to be dimers. You only need two subunits to make the full channel this time. So 1, 2 there. Okay, right, so now let's pull out a single one of those subunits, a single monomer basically, and have a look at its membrane spanning topology. Okay, so the reason then that these channels can assemble as dimers rather than tetramers is because each subunit effectively contains two of the subunits for inward rectifying potassium channels. Okay, so to make a functional potassium channel, you basically need 4 P loops that are lining this selectivity filter portion here. Basically, when we were building them out of these subunits that only contained a single P loop, therefore, we needed 4 subunits in order to make a full channel. In these two-pore domain potassium channels, each subunit is going to contain 2 P loops, and therefore effectively is equivalent to 2 of these other subunits in these other types of potassium channels. Okay, so let's draw this. So again, here is the cell membrane, and this is going to be the membrane spanning topology. So you're going to have the first membrane spanning alpha helix, the P loop, second membrane spanning alpha helix, the third membrane spanning alpha helix, another P loop, and then the fourth membrane spanning alpha helix, like so. So hopefully you can appreciate how effectively it's just like 2 inward rectifying potassium channel subunits stuck together. Okay, so let's have a look at the different structures here. So we have these 2 P loops here, which are both going to be lining the pore, okay, and therefore when you put 2 of these subunits together, you're going to end up with 4 P loops lining that selectivity filter again, so that's perfect. Okay, and then you have the 4 membrane spanning alpha helices, which I'll colour in orange, like so. Okay, right, so there are a lot of different subunits for two-pore domain potassium channels, just as there were for voltage-gated potassium channels, inward rectifying potassium channels, and calcium-activated potassium channels. And these are split into 18, well, not 18 actually, 15 different families, but the naming is slightly odd. Okay, so it goes from K2P all the way up to K2P18, but I've just said that there are only 15 families, and that's because there are certain families which are not present in humans. So the ones that are not present in humans are the K2P8 family, the K2P11 family, and the K2P14 family. Okay, so these 3 families are omitted basically, okay, and that's why there are only 15 families present in humans, although the naming seems to go up to K2P18. Okay, and again, the individual members of these families are named as before, where you have the family, so like, if I give you an example, K2P1, family number 1, and then you put point X, and this tells you the member of that family. Okay, now, before we move on, I should probably tell you what the function of these two-pore domain potassium channels is. Okay, so although they are the least famous of the potassium channels, arguably, okay, they are actually incredibly important. These are the potassium channels that you learn about in first-year physiology. These are the leak potassium channels, okay, that no one ever tells you actually what they are, but these are them. Okay, these are the ones which are always open in the membrane and which are always allowing potassium to leak out of the cell into the extracellular fluid, and that leak is utterly essential for the normal equilibrium potential across the cell membrane. Okay, so it's essential for establishing resting membrane potential across the cell membrane. Okay, right, so those are the two-pore domain potassium channels. Now, we're not going to see any examples of where these potassium channels are specifically involved in driving cancer, but when we have our more general discussion of how ion channels are dysregulated and that this leads to chronic depolarization of the electrical potential difference across the cell membranes of these cancer cells, these ones are potentially going to be extraordinarily important in that. Okay, so it's worth remembering this for when we do that. Okay, right, so that's the two-pore domain potassium channels. What we now want to talk about is channels other than potassium channels. Okay, so the other channels that I'm going to mention, because we're going to see examples of them as we progress through the videos, are voltage-gated sodium channels, voltage-gated calcium channels, and also a few trip channels as well I'll mention. Okay, right, so let's start off then with the voltage-gated sodium channels. So we're moving away from the potassium channels now, and we're going on to the voltage-gated sodium channels, which for short are abbreviated down to VGNC. So this stands for voltage, that's the V, and then gated, that's the G, and then Na for sodium, and then C for channels, voltage-gated sodium channels. Okay, right. So let me show you then the structure of the pore-forming unit of voltage-gated sodium channels. And I should just note at the moment that I have shown you the core structures of all of these channels that we have looked at. I have not shown you auxiliary subunits. Many of these channels end up having other proteins attached onto them, which are not essential for their function, but which can moderate their function and change their localization within the cell and things like that. Okay, so I'm only showing you the core structures, and again I will stick to that when I'm doing the voltage-gated sodium channels and the voltage-gated potassium channels. That's just because of time constraints. Okay, right, so voltage-gated sodium channels. Okay, right, so voltage-gated sodium channels, the core pore-forming unit of a voltage-gated sodium channel is just a single protein, okay? So they're much, much simpler than the other ones that we've looked at, because the fundamental pore-forming unit is just one protein, okay? So this single protein then is known as the alpha subunit of a voltage-gated sodium channel, and there are other subunits that can associate with it, known as the beta subunits, which are not essential for its function and therefore are auxiliary subunits, and we're going to omit them from our discussion. Now, despite the fact that it is just one protein that makes up this entire pore-forming unit, okay, it actually is split into 4 separate domains that resemble the 4 separate subunits of a voltage-gated potassium channel. Okay, so what we can do is split it up into 4 separate domains. Okay, domain number 1 domain number 2, domain number 3, and domain number 4. Okay, now let me show you the membrane spanning topology of this gigantic great protein. Okay, and I'll show you how it can be split into these 4 different domains. So we'll start with the amino terminus here. Okay, and then here comes the first domain here, like so, which you will notice is just like a single subunit of a voltage-gated potassium channel, and then it's linked by this looper into the next domain here, which is the second domain, which again is just like a single subunit of a voltage-gated potassium channel. And then here is the third domain here, which again is just like a single subunit of a voltage-gated potassium channel. And then finally a fourth repeat of this, like so. Okay, and then you finally end up with the C-terminus right over there. Okay, right, so let's highlight up the different bits here then. So this portion here, this is, well, actually I'll do this like so. This is domain number 1 here, and then after that you've got domain number 2, domain number 3, and then finally domain number 4, okay, here. Right. Then, within each domain, you then have the individual portions that resemble the structures within a single voltage-gated potassium channel subunit. Okay, so for instance, if we look at domain 1, you have the 6 membrane-spanning alpha helices, which are now in turquoise, okay, and those are labelled up S1 to S6, okay? So if you wanted to specify a specific membrane-spanning alpha helix in a voltage-gated sodium channel alpha subunit, what you would have to do is you'd firstly have to specify the domains. So you'd put D, and then you put 1, 2, or 3. So maybe, let's say we wanted to specify, just to pick a random one, let's say we wanted to specify that one there. Okay, we'd firstly say that was D3, to say that it was in domain 3, and then we'd say it was S, and then it's 4. Okay, so it's domain 3's S4 basically. Okay, so that's how you specify membrane-spanning alpha helices in the entire alpha subunit of a voltage-gated sodium channel. Okay, now we can also see that each one of these domains has a p-loop. Okay, and again these 4 p-loops will be surrounding the selectivity filter of the voltage-gated sodium channel. They'll be lining the pore on the extracellular side, and they are responsible for selecting the sodium ions over other ions. Okay, right, so that's the structure of the alpha subunit of our voltage-gated sodium channel. Now there are 9 different subunits, well there are 9 different alpha subunits for voltage-gated sodium channels. Okay, and these are named NAV 1, and then it's NAV 1.1, NAV 1.2, all the way up to NAV 1.9. Okay, so those are the 9 different alpha subunits of voltage-gated sodium channels. Okay, and again, they respond to depolarization of the electrical potential difference across the cell membrane, and when you depolarize the electrical potential difference across the cell membrane, they then open. And again, just like the voltage-gated potassium channels, you can split each one of these domains into the first 4 membrane-spanning alpha helix, which is the voltage sensing domain, and then the last 2 membrane-spanning alpha helices, S5 and S6, with the p-loop in between, which is then the pore-forming domain. Okay, right, and each one of those makes up 1/4 of the whole voltage-gated sodium channel alpha subunit. Okay, right, so we'll call it there for this video, and in the next video what we'll do is we'll discuss voltage-gated calcium channel alpha subunits, which are fairly similar to voltage-gated sodium channel alpha subunits, and then we'll discuss trip channels, and then we'll go on to looking at some specific examples of how they are involved in driving cancer, after looking at some general principles.